Hello everyone. As you can see from the course title, we will learn about microbiology, and in this lecture we will know what is microbiology and have a small talk about its history. So first, what is microbiology? Microbiology is the study of microorganisms such as bacteria, fungi, algae, protozoa, and viruses, in which viruses are acellular, meaning they are not alive. Microorganisms are the organism and entities like viruses that cannot be seen by the naked eye, meaning that we need a microscope to. After we simply defined microbiology and microorganism, let's have a brief look at how did this field started. As you can see, this photo, this is one of the greatest scientists ever. He is Robert Koch, who is an English natural philosopher, architecture, and polymath. He discovered cells and called them cells. Also, he drew them in his book Micrographia. From there, we got the first drawing of a microorganism, which was the fungus mucor. This is the first microorganism saw with Hooke's simple microscope. After that, scientists started to wonder about the diseases and where they came from. Second, the father of microbiology, Van Leeuwenhoek. He's a Dutch microbiologist. Saw a microorganism and described them accurately with his simple microscope composed of double convex glass lens held between two silver plates that magnify from 50 to 300 times. This is the microscope he designed. Let's take a laser. This is the microscope he designed. The specimen to be examined was placed on here. You can see it on here, on that needle. This is where you put the specimen. Then the specimen, you can put it into the position vertically by turning this screw here, this one. Then the specimen moved toward or away from the lens. This is the lens, by the way. This is the very small lens. We can move the specimen toward or away from this lens by turning this screw, This is the part of the microscope he designed. Then we have Louis Pasteur. He is French biologist, microbiologist, and chemist. He had a lot of contribution in microbiology since he and a lot of other scientists like Francisco Redi and Square rejected the spontaneous generation theory. Louis Pasteur did two experiments to reject this theory. One of them is this swan neck flask experiment you see right here. Okay? Pasteur placed a nutrient solution in this flask. Okay? Then he heated the neck and the flame and bended the neck of the flask out into curves to become like swan neck, keeping the end of the neck open to the atmosphere. Then Pasteur boiled the solution for a few minutes. That resulted in forcing the air out through the open end and then allowed the flask to cool down. Then Pasteur left the flask at room temperature for a long time. This is the incubation period. Then after the incubation, Pasteur broke the neck of the flask, exposing the nutrient solution to the open air, and kept the flask for a few days. What he saw after the experiment, growth appeared in the broken neck flask because microbes readily entered the flask via air and grew. He concluded that microorganisms are not spontaneously generated from non-living matter, but arise from other microorganisms. This theory is called germ theory. He discovered also the principle of vaccination. when he invented rabies vaccination, in which he take the pathogen from an infected rabid dog and injected it in a rabbit. When the rabbit died, he removed the brain and the spinal cord of the rabbit and powdered them, and then injected a 9-year-old boy called Joseph, who was bitten by an infected rabbit dog, by the dried material 13 times over 10 days, and, as he thought, the boy survived. The third thing he did is that he discovered fermentation, in which carbohydrates are converted to alcohol or organic acids. He found out that fermentation was carried out by living microorganisms, and this is a problem in wine industry. The wine spoiled, so he did pasteurization to prevent it by heating the wine to 55 Celsius for several minutes. To add, pasteurization is heating liquids to destroy microorganisms that cause spoilage and diseases. We have Robert Koch. He's a German physician and microbiologist. He demonstrated the relationship between bacteria and diseases, and also he developed Koch postulates. He found that Bacillus anthracis cause anthrax by isolating a microorganism from dead diseased animal, grow the microorganism in a pure culture and identify it. He injected a healthy mouse with the material taken from diseased animal. The mouse became sick. Koch isolated the microorganism from the sick mouse and transferred the anthrax bacteria through a series of 20 mice. After that, Koch incubated a piece of the spleen of the mouse containing the anthrax bacillus in beef serum. The bacilli grow, reproduce, and produce spore. Then Koch injected the growing bacilli into the mouse. The inoculated mouse developed anthrax. Then he put Koch postulates to show the relation between a microorganism and a disease, which where microorganism must present in every case of the disease, but absent from the healthy microorganism. The suspected microorganism must be isolated and grown in a pure culture. The same disease must result when the isolated microorganism is inoculated into a healthy host. The same microorganism must be isolated again from diseased host. Fifth, we have Hans Gram. He's a Danish bacteriologist who developed a Gram stain to differentiate between a Gram-positive and a Gram-negative bacteria based on the cell wall structure and make bacteria more invisible under a microscope. Gram-positive appear violet or purple, and Gram-negative appear pink or red. Sixth, we have David Burgess, who published a book called Burgess' Manual of Systematic Bacteriology in 1923 to determine the identity of bacteria using their characteristic. There's a lot of other contribution in microbiology by many scientists I didn't mention here. Search them and know more. And that's it for this lecture. See you next lecture. Thank you.